Welcome to this presentation on enzyme-modified electrodes. This cutting-edge technology combines the specificity of enzymatic reactions with the sensitivity of electrochemical detection. By immobilizing enzymes on electrode surfaces, we create biosensors that can detect even trace amounts of substrates with remarkable precision. As we delve deeper into this topic, we'll explore the mechanisms behind some enzymatic reactions, the types of enzymes commonly used, and the latest advancements in this exciting field. Today, we'll cover 5 key objectives. We'll start by exploring what enzyme-modified electrodes are. This section will cover what makes them unique compared to traditional electrodes. Next, we'll delve into how these sensors work. We'll then look at some sample data. This real-world evidence will demonstrate the performance of enzyme-modified electrodes in practical applications and experiments. Our fourth objective will focus on the strengths of enzyme-modified electrodes. And finally, we'll explore example applications of enzyme-modified electrodes. All right, let's dive into what makes these sensors so special. So what are enzyme-modified electrodes? EMEs are sensors whose surfaces have been modified with an enzyme, and this modification allows the electrodes to interact specifically with certain analytes, enhancing their detection capabilities. These sensors are particularly useful for detecting analytes that are inherently non-electroactive. By using enzymes, we can break down these non-electroactive analytes into electroactive byproducts, allowing us to detect these analytes through electrochemical techniques using redox proxies. The primary advantage of using EMEs is to monitor fluctuations of species that were previously undetectable using electrochemical methods. Some EMEs, such as chitosan modified microelectrodes, which are sold by Pinnacle Technologies, also had the ability to detect analytes in real time. The specific EMEs we'll talk about today have the capability of detecting both electroactive and non-electroactive species in real time, depending on the electrochemical technique you couple them with. This real-time detection is crucial in various applications, from medical diagnostics to environmental monitoring, where timely and accurate measurements are essential. Now, one might wonder, how do these sophisticated devices operate? The key lies in their selectivity. Enzymes on these electrodes specifically target their substrates and catalyze their breakdown into various side products. Oxidase enzymes, for example, produce hydrogen peroxide as a byproduct, which is denoted by its chemical formula H2O2. This produced hydrogen peroxide plays a pivotal role. Due to its electroactive nature, hydrogen peroxide acts as a reporter molecule. This means that when we are monitoring levels of molecules such as glucose, it's actually the hydrogen peroxide production we are measuring at the electrode surface. To visualize this process, let's look at the diagram at the bottom of the slide. Here we have our substrate approaching an oxidase enzyme. As they interact, several products form depending on the analyte of interest, one of which is hydrogen peroxide. As the hydrogen peroxide approaches the electrode surface, the electrode facilitates the transfer of 2 electrons, which can be measured as an electrical current. This current is directly proportional, for example, to the amount of glucose present in the sample, because higher concentrations of glucose will also lead to more hydrogen peroxide production. In essence, EMEs provide us with a powerful tool for detecting and quantifying biological molecules, such as glucose, through elegant biochemical reactions coupled with electrical signal output. Now, glucose is not the only non-electroactive substance that can be detected using an EME. On this slide, we're looking at some key examples of oxidase enzymes, or enzymes that catalyze oxidation reduction reactions using oxygen as the electron acceptor. First, we have lactate oxidase. Here you can see how lactate oxidase converts L-lactate along with oxygen to produce pyruvate and hydrogen peroxide. We also have glucose oxidase, as previously discussed, which is widely used in glucose sensors for diabetes management. It catalyzes the oxidation of D-glucose to form D-glucano delta-lactone, and hydrogen peroxide. Lastly, we have glutamate oxidase. This enzyme reacts with glutamate and oxygen to yield 3-oxoglutarate while producing hydrogen peroxide and ammonia. An important point to be noted is that each of these enzymes produces hydrogen peroxide as a byproduct that can be used as a reporter for the molecule of interest. This slide illustrates what these sensors look like at a microscopic level. On the left, we see the step-by-step process of preparing an EME. First, the enzyme is suspended in a chitosan solution. This mixture is then deposited onto the electrode surface. Through electrodeposition, the enzyme chitosan matrix forms a hydrogel that encompasses the sensor, resulting in an EME. On the right, we have several magnified images of the tip of an enzyme-modified electrode, in which the hydrogel is about 150 microns long. In my opinion, these sensors also have a wonderful resemblance to a certain carnival food. Delicious. Here we're presenting some empirical data illustrating how EMEs function in detecting glucose levels as we sweep from -0.2 volts up to 1.4 volts and back down. As we can see from the graph on the top left, when glucose is introduced to a bare electrode, there's no significant electrical response. This indicates that the glucose is undetectable to a bare electrode. However, if we use an enzyme-modified electrode, upon the introduction of glucose, we witness a sharp increase in current, peaking at about 33 nanoamps. This spike corresponds to an oxidative reaction where hydrogen peroxide is produced as a result of enzymatic activity. This clear distinction between responses highlights not only the sensitivity of EMEs, but also their specificity towards substrates like glucose, a crucial aspect for biosensing applications. Now we'll explore the unique strengths that make EMEs a powerful tool in electrochemical analysis. Firstly, one of the most significant strengths lies in the high selectivity enzymes offer. Each enzyme is tailored to interact with specific substrates, allowing us to target particular reactions with high precision. Secondly, these sensors are incredibly versatile. They can be employed alongside a broad spectrum of electroanalytical methods, making them suitable for various experimental setups and research needs. Lastly, some EMEs, like the ones we talked about today, can provide real-time insights into non-electroactive species depending on the techniques they are coupled with. This detection opens up new possibilities for dynamic analysis and monitoring that were previously challenging to achieve. Moving on to applications, let's discuss how EMEs are utilized in managing diabetes through blood glucose monitoring. There are two primary types of devices that benefit from this technology: instant meters and continuous blood glucose monitors. Instant meters like the OneTouch Ultra 2 shown here allow for quick measurements of blood sugar levels at any given time. Continuous blood glucose monitors offer real-time tracking of glucose levels throughout the day and night. Both these technologies represent significant advancements in diabetes care by incorporating enzyme-modified electrodes for accurate and reliable measurements. Thanks for watching.